All right, let's see here. Huh. Now, how do I? You guys can see my screen just fine?
Yes.
Okay, perfect. All right, so I actually really like this first image I'm showing you here. It's just some beeswax, some comb that had some fun lights behind it, and I felt like it really sets the tone for talking about heat tolerance in honeybees, with this kind of thermal red here. But I actually want to start off this whole conversation by talking about California in general and how California is a mystical bee place. And specifically, what I mean by that is um, we have commercial bee stocks in Northern California, and they're very important for pollination across the US because many beekeepers are relying on Northern California stock for their pollination. But in Southern California, these bees are shipped, not too far away. And when they're shipped here to Southern California, we noticed something kind of peculiar. Typically, these commercial queens, which is what I'm going to refer to, stock purchased from Northern California, these commercial queens have a lifespan typically of only about 6 months. And what um, we're noticing is that bees are purchased early in the year around springtime. They go through the summer, they experience several heat waves, and then they typically have some sort of crash or fail occurring in the fall. And we know that Varroa and the viruses that Varroa spreads are likely impacting the queen's fitness. But one thing that I've been thinking a lot about is how heat might be a driver of potentially infertility and queen loss. And essentially, the idea here is that we hear reports of bees or queens dying during heat waves. So there's something occurring in the biology of the bees where heat is affecting the queen. But I want to dive into that a little bit more later on as to what could be going on with the queen. I wanted to point out that where that little bee icon is where UC Riverside is located and where I do my research. And really, when it comes to honeybees and climate change, some questions that I want to know are, can we breed for climate resilience? Are there even populations that demonstrate tolerance to heat? And if they do demonstrate tolerance to heat, how are they achieving that? What are those underlying molecular mechanisms? And I'll get into those molecular mechanisms in a little bit here. And to add to the whole story that California is this mystical bee place is that we have two bees occurring in California. We have the commercial stocks in the north, but then we also have bees in Southern California that are um, just existing in the local landscape. Now, how they got there um, is its own story in and of itself, but essentially, um, there was a scientist back in 1956 in Brazil. His name was William Kerr, and he wanted to breed bees that were more resilient to um, the environment in Brazil, which is tropical and warm. And the European bees that they were using in Brazil were not well suited to that environment. So they actually brought bees from southern Africa to Brazil. And I think most of us probably know the story, but somehow they got out and um, the bees propagated in that environment and spread rapidly across the Western Hemisphere. And so we see those bees here in Southern California. But one thing that's really interesting is that they've been demonstrated to have really high genetic diversity. And so, genetic diversity we can think of as a bit of a biological insurance policy. It helps to, genetic diversity is essentially there when the going gets tough. Populations can rely on the genetics that they have. Maybe there are certain genetics that the bees possess, some of them that are really good in certain stress contexts. And so um, having a lot of genetics to tap into is really important for species and their persistence. And to give you another way to visualize what's going on with the bees in Southern California is that they are hybrids. And when we think about bees and their evolution, bees are native, or honeybees are native to what we would refer to as the old world. So, um, what we know about bees and their evolution in the old world is that there are major clades that these bees form in their breeding populations. Um, for example, Western Europe, Eastern Europe, these are your two clades that represent most of the what you would consider your European bees here in the US. So Western Europe bees are Apis mellifera mellifera, and bees in Eastern Europe, you can think of those as your Ligustica, your Italian honeybees are derived from Eastern European source. But we have also imported historically Middle Eastern honeybees into the US. And we know that African genetics have um, propagated themselves in the US as well. So, really, all of these genes are coming together and mixing into this one population, essentially. But bees in Northern California are also hybrids. They're just hybrids in a different way. They notably do not have the presence of the African ancestry there, but they are a mix of Western and Eastern Europe plus Middle Eastern bees. And I'll show you that a little later on. But I want to talk about honeybees and how they mate because it's important to understand their overall biology and what it means in the context of heat. So we start off with a virgin queen here, and a virgin queen is going to mate during a brief period at the beginning of her life. And during that brief period, she is going to mate with many, many males. That is called polyandry. So, honeybee queens are polyandrous maters. And the whole idea here is that they are sequestering sperm that they can store for the rest of their life. And queens can live for multiple years. Um, Often, or I wouldn't say often, but I think it's been noted that they can live for up to 7 years. So during that whole time, they're storing sperm from the initial mating period. But where I think that heat comes into play is that the queens, once they've mated, are in the colonies. A heat wave comes, and if the sperm inside of their spermatheca, which is their sperm storage organ, um, if that sperm begins to die due to heat, then the queens may have a compromised lifespan because queens do not live if there's no more live sperm in their spermatheca. So their life and their lifespan is directly dependent on the amount of live sperm that they're storing. So I kind of have this idea that heat is killing off sperm in the queens during heat waves. But another aspect is the male himself. And so this is an image of a male and his genitalia called the endophallus. And at the tip here, there's this goop. And this goop is the ejaculate. And that is a mix of sperm and seminal fluid. I think we're all familiar with the concept of sperm and what it does for biology and essentially just it's the sex cells of the male. But seminal fluid is maybe less known, but it's essentially a component of the total ejaculate that is coating the sperm and protecting it. And so this seminal fluid actually is really important. My advisor, Boris Baer, has studied seminal fluid extensively and has found a variety of proteins that are contained within this that do a lot of different functions. So, for example, there are neuropeptides in the seminal fluid, and these neuropeptides are affecting the queen and her brain function after mating, and they will blind the queen. And that's advantageous to the male because he wants to be the only father for the queen, but the queen wants to mate multiple times. So what does he do? He effectively blinds her so that she reduces her chances of mating. But bringing it back to the overall goal of understanding heat, I wanted to understand whether SoCal bees and commercial bees displayed any thermal tolerance. And so I looked at their survival rates, specifically of the drones, when they were exposed to heat. But then I also looked at the fertility of the drones when they're exposed to heat. And an interesting feature of drones is that unlike humans, and male humans specifically that are producing sperm, if a human gets stressed and that somehow depletes the live sperm in that male can continue to produce more sperm. That's not the case in drones. Drones produce all of their sperm for their entire life during their pupal stage. So when they emerge as adults, any stress that they face that impacts their fertility, they can't make up for that. So we can directly understand how certain stresses are affecting the fertility of males, which is pretty cool. So I conducted a common garden experiment, and so here is a look at the apiary that I maintained for my experiments. I really enjoyed being in this eucalyptus grove. And so essentially, I had both the SoCal bees and commercial bees mixed um, throughout this apiary, and this is called common garden experiments, where you rear them all under the same conditions. Then I took drones and nurse bees from those colonies and I put them into these 3D printed cages here. And I think this is pretty cool. You can see in the background the 3D printer, this orange apparatus here, and then it's actually printing off more of these cages, which are green right here. But then I provided all of the cages with a tube with honey and then another tube with water. And so essentially, I gave them food during their treatments, and the treatments were either 35C or 42C for 4 hours. So um, I know most of us probably don't work in Celsius. So in Fahrenheit, 42 degrees is about 107 degrees Fahrenheit. The reason why I picked these two temperatures is because we have data from sensors in our apiary that suggests that internal brood temperatures are around 35 degrees C. And then a typical hot day, or what we would get flagged for as a heat wave here in Southern California, is right around that 42 C mark. So essentially, we're trying to mimic internal brood temperatures where drones would be nice and cool, and then um, hot temperatures where drones might be flying out for a mating flight during these periods at 42 C. The other thing is that 4 hours was selected because we typically do not see males engaging in mating flights for longer than this period of time. So then after the treatments, we would squeeze the drones and extract their ejaculate. And you can see the ejaculate is right here. And we encase the ejaculate in some additional fluid so it doesn't dry out. But then I essentially gave all of the bees either a 35C or a 42C heat treatment for 4 hours. Some males, I looked at their fertility right after the treatment. And then some of the males I looked at 4 days later. So there's this short-term and long-term effect, and this becomes important. To quantify the fertility of the males, we use fluorescent staining of the sperm. So green sperm are live and red sperm are dead. So this is the field of view in the microscope is what this image is showing. And all of the green dots you're seeing are live sperm, and all of the red dots that you're seeing are dead sperm. So we're able to quantify a proportion of live to dead sperm. And I have to thank all three of these undergrads who helped me throughout this process counting sperm. They were in the microscope room under conditions where the dyes do not degrade, which means they're in complete dark because these dyes are light sensitive. So I really commend all three of these students for the hard work that they did. So first, let's look at the results for the survival. Here is a what's called a Kaplan-Meier survival curve. And how we will interpret this is that there is our x-axis here. And on the x-axis, we have days. So over the course of 4 days, I recorded how many males were dying. All colonies started off at 1 here. So the y-axis is your proportion that are surviving. So everyone at time 0 is alive. And then after 4 hours, I recorded their survival, which is what we see here. Then we see survival at day 1, survival at day 2, survival at day 3, and survival at day 4. And we have 4 different lines going on here as well. So the solid lines in both cases are your commercial bees, and the dotted lines in both cases are the SoCal bees. Orange indicates groups that were treated at 35C, and then the commercial or the red lines are for 42C. And what we notice is right at day 2, after the heat exposure, we're seeing this significant drop in survival for commercial bees when they're exposed to 42C. And we don't see that in the other groups. So this suggests that survival is affected for male honeybees in the commercial group exposed to heat. Now let's look at the fertility of the drones when they're exposed to heat. So here we have two different panels. This one on the left is the data at 4 hours, and then on the right, we've got the data for 4 days. And so we also have within one panel two different groupings going on. One is for the commercial group and all of the fertility measures that we got. The other is for the SoCal group and all the fertility measures. And so what we see in the 4 hour group um, is that, so the letters here indicate groups that would be different from one another. The groups all have an A above them, which means they're all the same group. They're not different from one another. So in the short term, we do not see an effect of heat on fertility in either group. And then in the long term, what we see is that the commercial group exposed to the 42C is experiencing a drop in their fertility. And many of the drones that we observed had completely inviable sperm. They were essentially sterile um, after 4 days when they were exposed to the heat. So that's quite alarming. But the other thing that's really interesting is that the SoCal group in both treatments is unaffected by the heat. It seems that they tolerate the heat just fine. It does not impact their fertility. So just to recap on the results I've shown you so far, SoCal drones are more likely to survive and they maintain higher fertility under heat. And then for my follow-up question, I was curious whether there are genomic or proteomic signals that are explaining this thermal tolerance. So here is a fundamental process in biology called the central dogma of biology. And what this is detailing is that we're looking inside a cell right now, and within that cell there is a core nucleus, and DNA is in that core nucleus, and it is getting transcribed into RNA. So RNA is essentially um, providing a code that the genes are providing. The genes are this blueprint. The RNA writes down the blueprint, and then translation occurs where the RNA is then synthesized, that blueprint, into a protein. So basically, the RNA says, arrange a protein in this way. And then the process of translation creates that protein. Really, the reason why I'm showing this is to demonstrate that genomics and proteomics are used as important tools to look at the ends of all of this. So at one end, you have the genomics, which is the DNA blueprint, and then the final result of the DNA is a protein of some sort. So I look at both of these perspectives, the genomics and the proteomics, to understand the honeybees in more detail. And so I wanted to break down really what the differences are in genomics and proteomics. And so for the question of what it is, genomics is understanding all of the DNA in the organism and essentially figuring out what its genetic blueprint is. But then proteomics is the study of all of the proteins in that organism. And we can think of proteins as phenotype. phenotype is the physical representation of an organism. We are not made of DNA. We're made of proteins. So um, when we talk about kind of a physical trait, it's something to do with the protein itself, not the genetics. For what these two things show, they're showing in terms of genomics what could happen. It's all of the genetic potential of that organism. But proteomics is telling us what's actually happening, the response of that organism to stress. So then, why this matters is that the genomics is helping us to identify the evolution of an organism, whereas the proteomics is revealing how that organism is responding to its environment, to stress, to disease. So it provides context for the genome. But there's limitations to both. So the limitation for genomics is that we don't know if genes are turned off or on. We just know that they're there. And if a gene is turned off or on, um, makes all of the difference in whether or not that we'll see a resulting protein in the organism. But for proteomics, proteins themselves are really susceptible to degrading. They break down very easily, and they're also susceptible to contamination. So if I am working with sperm and seminal fluid of an organism, I have to wear gloves because, for example, I might get my skin in contact with that. And then in my analysis of the proteomics, I'm picking up now uh, proteins that belong to me and not the organism. So um, proteomics is really sensitive to potential contamination. So I want to start with talking about the genomic aspects of the SoCal and commercial bees, and I really wanted to determine whether these bees are distinct populations or not. Are they just, the same thing? How do we really prove that they're different? So the first thing I did was collect samples of both commercial bees and SoCal bees, and I looked at their overall ancestry proportions. So each one of these bars represents a different individual, and each individual has some sort of ancestral makeup. And so we're able to look at how much of the organism's genome is contributed by X, Y, or Z ancestry. And so in this case, what we are seeing is that red is representing African ancestry, yellow is Eastern European, so those Italian bees. This light blue is the Middle Eastern bees, and black is for Western European bees. And what we see is that in both cases, commercial and SoCal bees are carrying about the same amount of both Middle Eastern and Western European ancestry. Where these bees start to differ is when we are talking about the Eastern European versus African. Notably, commercial bees, albeit a very, very small amount, which was probably negligible, um, is mostly this Eastern European. But in the SoCal bees, we're noticing that it's more of a balance or a push-pull between African ancestry and Eastern European ancestry. And what's really interesting is that we also detect a gradient to the African ancestry. So some individuals carry less than 25% of the of African ancestry in their genome, whereas others can carry close to 50%. So there's a range of African ancestry, and we would expect that to differences in your ancestry to shape your phenotype. So another way we can understand the genetics of honeybees is by um, displaying the each individual on a two-dimensional plane. So this is called a principal component analysis, short for PCA. And essentially, what we have is every individual that I looked at in this study and kind of where it exists in the universe that is the honeybee genome. And what we see is that Middle Eastern, Eastern European, and Western European ancestries are kind of more on the periphery of this. Um, in distance here is directly related to how closely related you might be to one another. It's another way we can think about this. So the fact that we see these things kind of spread more out means they're more distantly related, whereas the bees here in red are the African bees, and they're kind of overlapping with this purplish color, which are the SoCal bees. But they also exist, while there is a bit of overlap, they exist in distinct space. They're not completely overlapping, which is important. The other thing is that the SoCal bees start to overlap with the commercial bees. And the commercial bees are also closer in space to the Eastern European bees. So overall, where these bees are existing on this plot is it makes a lot of sense in the context of their ancestry that they have too. And then the last uh, piece of evidence I looked at was their genetic diversity. And I was mentioning this earlier, but genetic diversity is like a biological insurance policy. When the going gets rough, you can tap into that genetic diversity for more potential evolution and to adapt to your changing environment. So when I first looked at all of the nucleotide diversity. The shorthand name for nucleotide diversity is called pi. So I essentially measured pi for all, it's a population wide analysis, but I looked at this in chunks across the genome. So I essentially plotted the values of what all of these nucleotide diversity values were and compared them across all the populations. And so the letters here again indicate different um, groups. And so for across the genome, notably the African population had the highest genetic diversity. And this is something we've seen over and over again. But right behind the African population for genetic diversity was the SoCal population. So SoCal has notably very high genetic diversity across the whole genome. But then we've got other populations such as Middle Eastern that has more genetic diversity than commercial, but commercial has more genetic diversity than Western European and Eastern European. So overall, this kind of tells the story that when we just think about our two Bs here in the US, the commercial and the SoCal, there's more adaptive potential in that SoCal population. Now, I looked at all of the nucleotide diversity that was just occurring in um, genes associated with the heat shock response. And what's interesting here is that both the African and Southern California population have the highest genetic diversity. So they are in one group together. There's no difference between them in their genetic diversity in heat shock genes. But the commercial population is um, has lower diversity again in heat shock SNPs, but maybe not as low of genetic diversity as it does genome wide. So what I'm saying here is that um, the commercial bees and Middle Eastern bees kind of form this second group of genetic diversity in heat shock SNPs. And then again, your Western European and Eastern European are in last place in terms of overall genetic diversity that they have in those heat shock genes. To recap on what we've learned in the genomic section of this, both SoCal and commercial bees are distinct hybrid populations. They are not pure ancestries like we see in their old world populations. But here in the US, these bees are essentially mutts, kind of like dogs, I guess. We've got our purebred lines, but then we also just have the uh, dogs that are a mix of everything. And what do we know about, or those of you that are dog lovers in the crowd probably know that mutts tend to be much more resilient and healthier than purebred dogs are. And it's kind of the same concept with bees, too. The other point I wanted to make is that those SoCal bees have higher genetic diversity, and that suggests that there's greater adaptive potential here. For the proteomic portion, I wanted to compare sperm and seminal fluid expression in both SoCal and commercial bees. So when we looked at the amount of proteins that were expressed, this is these four panels here tell you about all of the protein expression and proteins that we collectively quantified. So in this top corner here, we see that in the sperm, we identified a total of 2,888 proteins. Out of those, only 1,157 were actually quantified because we set certain criteria for what proteins will actually compare to one another. First of all, they have to be a protein that's present in both groups. So these Venn diagrams are kind of showing that there were 700 proteins that were only identified in the sperm of Southern California bees, whereas in the commercial bees, there were 62 proteins that were only found in those bees. But they overlapped in about 2,123 of them. But then we further filter that because we have to set a threshold that over half of all of the bees um, have that protein present. So that reduces it down even further. Um, so of these 1,157 proteins that were quantified, 346 of them showed DEP, which is differentially expressed protein patterns. So essentially, there were 346 proteins that were at either a higher or lower level, uh, significantly from a statistical standpoint, they were higher or lower than the commercial group. But then in the seminal fluid, there were only three proteins that were differentially expressed. So these bottom panels here are just showing within SoCal, there's sperm and seminal fluid proteins, and there is overlap. There's some differences, but also overlap in the proteins they contain. And many of them, though, are differentially expressed. Then when we look at the commercial bees, it's a similar story where um, there's a lot of overlap in the proteins found in both the sperm and seminal fluid, and quite a few of them are also being differentially expressed. But overall, it's what became interesting to me was comparing the SoCal and commercial bees, and these 346 proteins in the sperm uh, seemed to have a story to tell, or they wanted to tell a story. So I decided to investigate them further. And so what we're seeing here is, I know it could maybe look a little scary, but essentially we've got the protein expression profiles of um, our bees that we sampled. So these pink colors are bees that were from the SoCal group, and then the green colors here are bees that were in the commercial group. So we see a chunk of green here, um, and each little square, you might be able to see that, corresponds to the proteins found in that one individual. So these are all the proteins for this one individual, and they go down in a list here. And the red indicates that the protein was really highly expressed, whereas the blue would indicate that this protein was um, expressed at a much lower level. So really this panel A here is just showing that um, there is within the commercial group, there's this whole chunk of proteins that are being more highly expressed. Then when we look at the pink, we're seeing that those that were really highly expressed here are much lower expressed here. So there's differences in the proteins that they are producing in the sperm. And these proteins are carrying out various biological functions. And we essentially, um, proteins can collectively be performing certain tasks. And so we call these gene ontology terms. So you'll see that this is labeled GO for gene ontology. And so essentially, um, we have these um, within this total protein expression profile here, we've got proteins that are involved in certain processes. And it's significant that these proteins are being differentially expressed and performing this function essentially. I won't get too into the weeds onto the technicalities of it, but just um, follow me on this whole adventure that there is this function called protein folding chaperone. And there were a lot of proteins that perform protein folding chaperone um, that were being differentially expressed essentially. And when we focus in on these protein folding chaperone, they're all heat shock proteins. So even though we didn't heat shock the bees, they were innately um, having these differential expression patterns in heat shock proteins, which was really peculiar. So essentially, we've got in green the commercial, and in pink is the SoCal. And each of the stars here indicates that this protein, heat shock protein, was differentially expressed between the two. And notably, across all of these heat shock proteins, they're being expressed at a higher level in the commercial bees compared to the SoCal bees, which was really interesting and not intuitive because our initial idea was that, oh, the SoCal bees are going to just have really high expression of heat shock proteins because they are, naturally evolving in this really hot climate in Southern California. But that wasn't the case. So that's kind of a fun part of science is when you have one sort of prediction about how the data is going to look out, but then you actually get the data and it tells you something else. Um, so in the sperm, we had this pattern of differential expression occurring, but in the seminal fluid, we um, look at this again, and none of these heat shock proteins were being differentially expressed like they were in the sperm. However, there is this one heat shock protein called protein lethal 2 essential for life, very long name. We abbreviate it as this PL2EL. Um, it was not expressed or found at all in the sperm. It was found in the seminal fluid, and when we find it, it's being more highly expressed in the commercial bees compared to our Southern California bees. So there's this really interesting differential expression pattern going on. And so I want to talk more about heat shock proteins and what it means to be a chaperone protein, because I'll go back. I was talking about this protein folding chaperone being a really important function, and hopefully this is making a little bit more sense now. But essentially, when a cell, which is what we're seeing in this blue here, encounters stress, maybe that is a heat shock, maybe that's an oxidative stress, maybe it's a mechanical stress, but something that is harming the cell. um, the proteins within that cell may be compromised. And so the shape of a protein is really, really important for its function. So this protein right here is misshapen, but these chaperone proteins, chaperone being a helper protein, they come in and they can assist in helping a protein to fold properly. They can assist in a protein that needs to be degraded because it's too misfolded to be reshaped. So they assist in degrading the protein if it needs to be degraded. They're essentially doing all of these little kind of housekeeping roles, cleaning up the cell and making sure that it's operating optimally. And so,
And just a quick time check, around 10 minutes left.
great,
Yeah.
perfect. Just about to wrap up. So, kind of in the context of what these chaperone proteins are doing and why I think there might be higher expression of these heat shock proteins in the commercial bees is potentially that the heat shock proteins themselves in commercial bees are not as efficient. So, you need more of them in order to function properly. And so, kind of to visualize this better, imagine that we've got the SoCal bees, and this is a protein that needs folding. Well, maybe it only needs two heat shock proteins to fold that protein properly. Whereas in the commercial bees, it needs a whole bunch of heat shock proteins in order to achieve that exact same function. So, overall, I just wanted to recap on what we've found out with the proteomics is that there are really dramatic differences in protein expression, but the most dramatic of these differences are occurring in heat shock proteins. And my current hypothesis is that the SoCal bees may just need to produce fewer of these heat shock proteins because the proteins that they have are more efficient. And to conclude overall, and what we've learned over the course of this lecture is that SoCal bees are displaying thermal tolerance as indicated by that heat shock assay in the beginning. And these populations are different genetically speaking, and the most prominent difference in their genetics is that presence of the African genetics. And so these African genetics in the SoCal bees are likely shaping both their heat shock expression and tolerance profiles. But the question becomes, can we breed commercial bees to be thermal tolerant? And that becomes a big challenge because the commercial bees themselves don't display innate thermal tolerance, so they likely would need breeding from bees that have thermal tolerance, like Southern California bees, to gain such a trait. But we know that bees, all Africanized bees, are more defensive. And so that creates its own challenge. If we want thermally tolerant bees, we have to figure out potentially how to bring these genetics in without bringing in those traits that are less desirable in these bees. So that becomes kind of the grand challenge with breeding for thermal tolerance. And with that, I'll take any questions. Thank you all for listening. It's been really great to give this lecture.